Assalamualaikum and good day everyone. So this is our research presentation, which the title is Rapid Forensic Differentiation of Human and Animal Bones using handheld NIR spectroscopy and deep learning. So my name is Nurul Afika Nabila Ahmad Zahar. My group member are Izzah Shafiyah, Roslan and also Nur Khalidah Halid. Next, I will continue with the background on the problem statement. So, in the study, we focus on the problem of identifying bone fragments in forensic investigations. So, in many real cases, the remains recovered are often burnt, degraded, or highly fragmented, which makes a traditional identification methods more difficult. So, the common techniques such as morphological comparison, histology, DNA analysis, and also the FTIR spectroscopy are useful, but they come with limitations. Because these traditional methods are slow, time-consuming, and in some cases it disruptive to the sample. They also require a trained specialist and proper laboratory facilities. So we choose an NIR spectroscopy because it takes only seconds per measurement and does not damage the bone, and it also comes with a portable which can connect to the smartphones, making them more practical for on-site forensic work. So this is a video of how the NIR works. The aim of this study is to evaluate whether a handheld NIR spectrometer can identify a bone samples for forensic use, and we develop an artificial neural network or ANN models to classify both human and animal bones and to see whether NIR can detect species differences as well as changes linked to post-mortem interval. So overall, the goal is to propose a fast, portable and non-destructive method for real-time forensic bone analysis. For this study, we analyzed a total of 225 femoral bone samples. So the femur was chosen because it has a consistent structure and reduced anatomical variation, which helps improve the NIR accuracy. So from these, 99 samples were human bones taken from real forensic cases. They were grouped into 5 postmortem interval classes, and we also include the medieval archaeological bones to extend the BMI range. So for the animal dataset, we collected 121 samples, which is from cattle, pork, red deer, and camels, which is these species were selected because they are commonly mistaken for human remains in forensic. This slide explains the methodology used in our study, which is we use NIR instrumentation. So we use handheld device, which is operating in the 1550 to 1950 nanometer wavelength range, and the device is about 138 grams, which suitable for the field-based forensic applications. So for the data collection, we use 8 identical handheld sensor, which bone sample was scanned 40 times on each sensor, producing a total of 71,720 spectra. So this large dataset improves the reliability and robustness of the machine learning models. So for the data analysis, we develop 2 types of classifier, which is a binary classifier for human versus animal, and the second one is multi-shade classifier for 5 species to differentiate a human from animal bones.
Hello, my name is Nur Izzah Shafiyah, and I'm going to continue the presentation to discuss the result from the research development. So, the study used a handheld near-infrared NIR spectroscopy to collect the spectral data from bone surfaces, and the classification decision was generated by a trained model called the artificial neural network ANN classifier. So the researcher analysed how the spectra of human can differ with the spectra of animal. So both of human and animal bone spectra are shaped by their molecular composition, but the absorption intensity and peaks vary depending on relative amount of collagen and exposed mineral. Hence, each class will show different band intensity and clustering behavior after pre-processing. So starting with the binary classification, the model was trained to distinguish human versus animal bone, and it showed an excellent performance. So the system achieved an accuracy of 96.3%, meaning almost all bones were classified into correct category. and the precision was 97.6%, showing a very low false alarm. As you can see from the spectrum, human bones resulting in broader and more intense absorption band compared to the animal bones. The result shows that the differences between human and animal spectra was clear for the model to draw a strong line and differentiate between the human and animal bone. Next is the multi-class classification, which it was expanded into 5 categories: human, cattle, chamois, deer, and pork. So compared to the previous binary classification, the overall accuracy for the multi-class classification dropped to 77.8% and with average precision of 58.6%. So based on the spectra, pork bone spectra formed the most distinct cluster and were the easiest to be recognized by the model. Deer also showed a strong classification with 95% correct identification. However, there was noticeable confusion between human and cattle bone because both come from large mammal and dominated by hydroxyapatite, hence producing the overlapping NIR absorption signal within the spectral range. So the model misclassified 45.8% of cattle spectra as human, but it dropped significantly to 9.4% when re-evaluated after pre-processing technique to remove the unwanted noise from spectral data. Lastly, for PMI analysis, the results show that human bone spectra change noticeably as the PMI increases, especially in class 5 where the PMI of the bone is over 100 years. So based on the PCA results show that the class 5 bone separated the most from bone from other classes, which prove that an IR signal can detect the change of chemical composition such as collagen degradation and mineral exposure over time. So, based on the result, there is a possibility that NIR may also help in estimating the PMI in the future. For the practical application of this research, it was demonstrated that the NIR spectroscopy is extremely lightweight with about 200 to 300 grams, which make it easy to carry and operate directly at crime scene or any remote forensic location without requiring the laboratory infrastructure. So one of the major advantages that it can scan the bone surfaces within seconds to allow for quick decision making. So the method is also fully non-destructive, meaning the bone sample can remain intact after scanning and preserving its potential for DNA analysis or other forensic procedure that may follow. So in terms of real-world practicality, this tool is especially useful in scenario like preliminary screening during mass disaster for victim identification and supporting archaeologists for bone screening.
Hi, my name is Nur Khalida Halid, and I will be continuing the presentations with the limitation. Although the study shows strong potential, several limitations need to be considered for real forensic use. First, the handheld NIR device only covers a small spectral range of 1550 until 1950 nanometer. This means it cannot capture important signals above 200 nanometer that relate to collagen, water, and mineral content. Missing these features can reduce the model's ability to tell apart species with similar bone chemistry, like humans and cattle. The study also used only femoral diaphysis samples, which are very dense and highly mineralized. Other bones, such as ribs, skulls, and vertebrae, have different structures and porosity, which may produce different spectral patterns. Because of this, the model cannot yet be assumed to work for the whole skeleton. Another major limitation is the absence of dog and cat samples. Even though these species are commonly found in forensic cases, especially in Malaysia, without them, the model is less useful when trying to quickly tell human remains apart from domestic animals. The multi-class deep learning model also struggles with species that have overlapping spectra. This is especially true to humans and cattle, which share similar long bone structure and mineral organic ratios. Finally, all bones were cleaned in the lab before measurement. In real situations, bones may be dirty, weathered, burned, buried, or decomposed. These conditions can affect NIR reflectance, meaning the model may not perform the same way outside controlled laboratory settings. To make this technique more reliable for forensic work, several improvements can be made. First, future datasets should include more species, especially dogs and cats, since they are commonly encountered in real investigations. Adding smaller mammals or poultry would also expand the model's usefulness in different forensic environments. The study should also include more types of bones. Real cases often involve fragments of ribs, skulls, or vertebrae, not just femoral shafts. Including the bones will help the model learn how different bone densities and structures affect the spectrum. Technically, accuracy could be improved by using NIR devices with wider spectral ranges, such as 900 to 2500 nanometer. A broader range captures more chemical information. Another useful step is combining NIR with other methods like Raman or mid-IR spectroscopy to create a more complete chemical profile of the bone. Lastly, the model should be tested under real forensic conditions. This includes bones that are burned, buried, weathered, or decomposed. Environmental factors can change the spectral signals, so testing real-world samples is crucial for confirming practical reliability. Overall, handheld and IR spectroscopy combined with deep learning offers a rapid, portable, and non-destructive approach for distinguishing human from animal bones. The binary model achieved high accuracy of around 96%, showing strong potential for quick, on-site forensic screening. While species level classification is still limited, the method lays an important foundation for more accessible forensic bone identification. With expanded datasets, improved spectral coverage, and more advanced AI models, this technology has the potential to significantly enhance real time forensic investigation and support faster, more efficient decision making in the field. I think that's all from us. With that, we thank you.